Hello, everyone. My name is Carolina Parshik. For my Office of Integrity Misconduct presentation case, I will be talking about the misconduct done by Logan Fulford. A little background about Logan. He was a graduate student attending the University of Cincinnati and worked at the Cincinnati Children's Medical Center, where he was a graduate research assistant. Later on in life, he became a senior clinical research associate at IQVIA. Furthermore, he focused on prostate cancers and finding some research about it. There was no known causes of prostate cancer. There are a few risk factors that causes prostate cells to become cancerous cells. Overall, prostate cancer is caused due to DNA mutations, and over time, our DNA, as it is the chemical makeup of our genes, which controls how our cells are formed and how they function. DNA mutations occur in a person's lifetime. Most gene mutations related to prostate cancers develop in a man's life and is not inherited. Male hormones, such as testosterone, promotes prostate cell growth. With certain risk factors, it can lead to an increase in levels of hormone, which can contribute to prostate cancer. Some certain risk factors that I did find was that older age, race, family history, and obesity could be indicators to prostate cancer. Logan Fulford research summary. Specifically, what Logan had focused on was prostate tumors, and he looked at it from the inside out, meaning that he was looking at individuals' prostate tumors through an expression of genes. And specifically, the one gene that he did find was the encoding the transcription factor of pork head box F1, which is typically not expressed in normal prostate epithelium. Logan Fulford primarily focused on using mice to see the effects of the overexpression of FOXF1, finding that there was an increase in tumor growth and metastatic progression. There was a detection of FOXF1 positive tumors in mouse and human PCA. They used murine off-topic models of PCA, finding an overexpression of FOXF1 in MICAP and TRAP prostate tumor cells, inducing tumor growth. Logan Fulford did conduct and participate in research misconduct, meaning that he did falsify data and had a data fabrication. The U.S. Office of Research Integrity did investigate Fulford, finding that while working at the university supported by 2 federal funds granted, he did engage in research misconduct. According to ORII, he was intentionally, knowing, and recklessly falsifying data. By doing so, what he was doing specifically was relabeling and reusing images for his own findings. Shown here was one of the 6 figures that were falsified in his journal. And while they were doing the investigation, they did find that out of those 6 that there was relabeling and reusing for his own findings. And the paper was later retracted in 2018 from Science Signaling Paper. ORII found this information acknowledgeable, and Fulford willingly falsified this data. In the figure shown below, it was one of the 6 that, as I did mention, and it was a Western blot that he did falsify. And so pretty much what he did was represent the expression of multiple different proteins under different experimental conditions while he was conducting the data, so it would be more beneficial for him. ORII's response overall was that he was guilty for the fabrication and falsification, and his article did get retracted in 2018. The response and sanctions were that Logan Fulford had to come to an agreement as to how he's going to be monitored when he was doing any type of federal research. So he didn't lose his status in conducting any type of research, but he had to be on supervision for about 2 years, beginning the 8th of May in 2020, as he became to come to terms with the agreement and tried to design a way of interacting and respecting the ORI's requirements. The guidelines were in place just to ensure that he was following scientific integrity. Lastly, the scientific impact that this could have led to was that if there was proper research that was conducted, it would have made a great impact for men who are struggling with prostate cancer. As mentioned prior, there is no known cause, and if we did find a way of finding how and why we can stop these tumors from growing. Logan's research could have been effective, but due to data falsification, it was not effective whatsoever. And these are my references.